Hello, everyone. It is my pleasure to introduce the ArQule management team, and they will be talking about small molecules in precision medicine.
Thanks, Gil. Good morning, everybody. I'm Peter Lawrence. I'm the president of ArQule. Thank you for coming to the presentation, and Gil, thank you for having us at the 18th Annual Needham Healthcare Conference. Before I begin, well, since I figured out how to work this, find the right communications device. There we go. Before I begin, just point your attention to the forward-looking statement slide. We will be making forward-looking statements, and the presentation contains forward-looking statements within the meaning of the Private Securities Litigation Reform Act. Actual results may vary materially from these statements based on risk factors contained in our periodic reports, including our recently filed 10-K. For those of you who have followed ArQule, we are an experienced developer of novel kinase inhibitors for precision medicine. Currently, we have 4 oral compounds in clinical development, all in biomarker-defined patient populations. The first of these, which is attracting a lot of attention recently, is a reversible BTK inhibitor called ARQ-531 that has been designed to overcome ibrutinib resistance. For those of you who follow irreversible inhibitors like ibrutinib and acalabrutinib, eventually patients become refractory to those molecules or those drugs as a result of BTK mutating at the C481S cysteine, and so you get what is called the C481S mutation. Our drug is a potent inhibitor of both wild-type and C481S mutant BTK, so we can effectively treat patients who have become refractory to ibrutinib. Our second molecule is an AKT inhibitor called mirinsertib. It is about to enter a registrational trial in overgrowth diseases, both Proteus syndrome and PROS. So we'll have more on that in a moment. Our third drug in clinical development is a next-generation AKT inhibitor called ARQ-751, and this is really designed for oncology. It is the most potent AKT inhibitor biochemically extant. It has excellent drug-like properties, and it has a broader therapeutic window than our first-generation AKT inhibitor, which is important because it is an oncology drug, and we're therefore able to dose it up to therapeutic levels. And we're actually seeing responses at levels where we're not seeing toxicity, which is actually unusual for AKT inhibitors. The other reason this is important is some very large pharmas have started to explore AKT inhibitors in hormone-sensitive tumors. We're in the process of doing that as well, and we'll talk a little bit more about that later. Finally, our fourth drug is an FGF inhibitor called derazantinib. Derazantinib is in a registrational trial in intrahepatic cholangiocarcinoma with FGFR2 fusions. This drug has been partnered in most of the world with a company called Basilea, and in Greater China with Sinovant Sciences Limited, which is a subsidiary of Roivant. So that's the portfolio. Moving on to the next slide, you see a summary of our program as well as pipeline goals. So returning for a moment to our reversible BTK inhibitor, ARQ-531, it is currently in a phase 1A/B dose escalation study. We're fairly far along in the phase 1A portion. It's attracting a lot of attention because it is, we're looking at the C481S mutant population, this biomarker-defined population. We have seen excellent drug engagement, excellent target engagement. We've seen very, very good PK, and we have seen excellent response based on sort of a linear drug dosing. Most importantly, and recently, we announced the first PR in cohort 7 of this phase 1A trial. It was a deep PR, about an 88% response, and that makes this, I believe, only the second reversible inhibitor that has seen a PR in the C481S mutant patient population. We feel now that we're getting very close to a therapeutic dose. We hope to wrap up the phase 1A and begin the phase 1B expansion cohort soon, and then if you look at the next, the The next target for expansion, it would be a fast-to-market registrational trial in the C481S mutant population. This is a very, very large market. It's a blockbuster-sized market in CLL alone, and we're excited about moving that forward. Next, our AKT inhibitor mirinsertib. As I mentioned, we are about to launch a registrational trial in Proteus syndrome and PROS. We have orphan designation. We have rare pediatric disease designation in Proteus syndrome, which means if approved, we would get a rare pediatric disease voucher, the most recent one of which sold for $105 million about 2 weeks ago. And we also have fast track designation in PROS and an opportunity to apply for breakthrough status in the not too distant future. The outcome of this work should be an NDA filing in either or both of Proteus syndrome and PROS. You'll see we describe the market as meaningful aggregate prevalence. By that we mean, although Proteus syndrome is a fairly small ultra-rare disease, PROS is larger. Together they represent several thousand patients, and that is not a trivial size for an ultra-rare indication, particularly with the way those drugs are priced. We're also taking mirinsertib forward in one phase 1b trial in oncology. It's in advanced endometrial cancer patients. I mentioned a moment ago that major companies like Roche are spending hundreds of millions of dollars on trials now in hormone-sensitive tumors with their AKT inhibitors in combination with both chemotherapy and also with hormonal therapy. And this drug, or our drug, is in phase 1b in combination with the hormonal therapy anastrozole. Turning to our next generation inhibitor, ARQ-751, that is in a phase 1b expansion study. We completed the phase 1a recently. We chose a recommended phase 2 dose. As I mentioned before, we saw efficacy without significant side effects. We're excited about that. That is not common with AKT inhibitors. We think this drug has the potency and the therapeutic window to be successful in oncology. We will be looking at opportunities in phase 2 later this year. A likely choice might be a breast cancer trial, as we've already seen 2 single-agent PRs with this drug in breast cancer. Next, derazantinib, the FGFR inhibitor. As I mentioned, it's in a registrational trial with orphan designation. That trial right now is being run by Basilea, our partner, and the outcome of that trial, if successful, will be an NDA filing. Moving on, I'm just going to focus, because we've covered some of this, I just want to focus on 2 things. If you look at the middle of this slide, you see IP protection. As you can see, all of our composition of matter patents go into the 2030s, with our BTK inhibitor, ARQ-531, going until December of 2035. We think this is quite relevant from a commercial standpoint. And now turning to key 2019 catalysts, focusing again on ARQ-531, we're hoping to select a final dose, a recommended phase 2 dose relatively soon, commence the enrollment of the expansion cohort, and plan for a fast-to-market registrational trial in patients refractory to ibrutinib or other frontline treatments that have the C481S mutation. So we're very excited about what's coming in 2019. For mirinsertib, as I already mentioned, we're about to commence the registrational trial in both Proteus syndrome and PROS. The next catalyst for mirinsertib in oncology would be the presentation of the data in endometrial cancer. With respect to our next-gen AKT inhibitor, ARQ-751, we intend to present data from the phase 1b study later this year, and then prioritize further development, as I said, likely in a phase 2 setting and possibly in breast cancer. And then finally, with respect to our FGFR inhibitor, Basilea is already expanding from the one trial in intrahepatic cholangiocarcinoma, and we expect that they will be announcing other expansion trials soon. And Sinovant is, we hope, about to initiate clinical trials in China. So that's a high-level overview of our portfolio and what we're trying to achieve this year. And now I'm going to turn it over to our CFO, Dr. Mark Sedgeran, who's going to take you through some of the details of these programs. Mark.
Thanks, Pete, and thanks very much for having us. My name is Mark Sadrian. I'm the CFO and Head of Strategy at ArQule, and I'll walk you through a little bit of our foundational science and some of the clinical data that we've seen so far at ArQule. So starting with ARQ-531, again, Pete touched on it, but there's a huge unmet need in CLL that remains. So obviously there's been tremendous advances that have been made in the field over the decades, but even over the last couple of years with the emergence of ibrutinib in particular first, but also venetoclax. And so these patients often do very well initially, but unfortunately, Clever being cancer has invented a workaround, in particular to ibrutinib's covalent binding to the cysteine residue at position 481 in the BTK molecule. And so as a result, the prevalence of that mutation and the number of folks who are both starting on ibrutinib initially and even experiencing very good effects for sometimes years, unfortunately fall off a cliff, if you will, very rapidly after the emergence of that mutation. So there's a very large unmet need given this large and growing market potential in CLL in particular, but also more generally in B-cell malignancies broadly, which for the most part are driven by amplifications of the BTK signaling axis. So just getting into the mechanism, you can see there's a lot of overlap between the way our drug binds on the right, ARQ-531, and the way ibrutinib binds on the left. Both bind quite tightly into the BTK binding pocket and prevent the phosphorylation of BTK at the site kind of at the top right of those pictures. One big difference, as you can see, is that ibrutinib binds covalently to the cysteine residue at position 481. One of the attributes of our drug, our molecule, that was required for taking it forward out of preclinical studies and into the clinic by our principal investigator was that our molecule is relatively agnostic to the presence or absence of that mutation at position 481. As you can see on the right, our drug binds near to but does not interact with that amino acid at that point. And so whether the mutation is there or not, our drug is relatively indifferent to. As you can see from the Western blots all the way on the right, both molecules do an excellent job of inhibiting the phosphorylation of wild-type BTK, the two green boxes. And then on the right, in the presence of the mutation, you can see that ibrutinib does not, and ours does. This essentially is the fundamental mechanistic premise underlying the development of this drug. Just looking at some of the in vivo mouse model data, this is a TCL1 CLL mouse model. In the middle trial, you can see placebo mice had all died by around day 40. All the ibrutinib mice had died by around day 60. And at day 74, it's not written there, but treatment ending is on day 74. More than half of the ARQ-531 treated mice were still alive, so median survival was not reached, showing a dramatic difference between ibrutinib, but also, of course, placebo in these mice. Going into the clinical trial design and data over the next couple slides, a lot of our mechanistic rationale and sort of preclinical data are now being trumped by the emergence of a more clinical profile, if you will. So right now we've cleared the first 7 dose cohorts for safety. That's from 5 to 65 milligrams. And we're currently enrolling cohort 8, which is 75 milligrams POQD. We've been announcing up to the date data for this ongoing dose escalation trial over the course of 2018 at AACR, at EHA last June, and then at ASH in December, and I'll show you some of that data. The only additional data that's been presented was at our 10K call several weeks ago, and I'll go through a little bit of that as well. So getting into the AEs and PK, again, very highly predictable PK, and we're getting into very substantial exposures now as we go from 45 to 65 milligrams that certainly should be clinically relevant. At the time of ASH, this was our AE profile. The only material update I would add to this very benign profile that was shown at ASH is that in cohort 7, as was reported a couple weeks ago, we did have a DLT in the form of a grade 3 rash at 65 milligrams. So that dose cohort was expanded and it was not repeated, and that's why that dose was cleared for safety. We hadn't seen that DLT in the past at lower doses, but of course we'll keep an eye out for that in the future. Getting into the more interesting slide here with respect to our pharmacodynamic effect and clinical data. This was data that was presented at ASH, and likewise we had an update at our 10K call a couple of weeks ago. So on the left hand of the slide you can see our pharmacodynamic data, and what it shows essentially is a little bit of phospho-BTK suppression at the very lowest doses. But once you get over 20 to 30 milligrams, you see essentially 100% inhibition of the BTK signaling axis and phospho-BTK. This correlates very nicely with the clinical effect on the tumors that we saw. So on the right-hand side of the page, you can see by dose cohort, lighter being the lowest doses and darker being the highest doses, what we saw in terms of tumor reductions, tumor volume reductions in these patients. As you can see from the chart on the right, there's a very sort of, I would say, clear dose response with respect to the highest doses achieving the largest decrease in tumor volume. In fact, those two patients on the right were both dosed at 45 milligrams. No 65 milligram data was available yet, and those patients had only received one scan. So they achieved a 40% tumor burden reduction at just the first scan at 45 milligrams. Another update that we made, as I alluded to, is that one of the patients, the first CLL patient who was scanned at 65 milligrams, achieved an 88% tumor reduction at the first scan. We also said that we'd be announcing more data from cohort 7, all of the data that we have, at a conference this summer, most likely EHA. So just getting into sort of next steps for us this year and beyond. So as Pete mentioned, we'd like to identify a recommended phase 2 dose. We think we're at or very near doing that in terms of dose and also in terms of time. At that point, we can start to initiate our expansion cohorts in most obviously a C41S, ibrutinib refractory population of CLL patients, but also other B-cell malignancies. Richter's transformation is another very fast-to-market pathway for us, and we've seen some preclinical efficacy suggesting that we have activity there as well. Understandably, it's a very tough disease where we haven't seen successes with other drugs, but given the unique mechanistic profile and kinase inhibition profile of this drug, we think there's a good chance there. So that would be another example of an expansion cohort we could run. So moving now into moransertib in rare overgrowth spectrum disorders. We are targeting, as Pete mentioned, two distinct but related overgrowth syndromes. They're both very rare. They're both caused by a somatic, sporadic mutation during early embryological development that leads to a gain of function and hyperactivity of the AKT signaling axis. This can either be a gain of function mutation in AKT itself or in PI3 kinase, which codes for the PIK3CA, which codes for the alpha catalytic subunit of PI3 kinase. In either case, the results are very catastrophic, and our program is designed to interrupt the signaling cascade in both of those conditions. So our first-generation AKT inhibitor, moransertib, has a dual mechanism of action. It binds to both inactive AKT as well as membrane-bound activated AKT. So Proteus syndrome, starting with Proteus first, again, it's a gain-of-function mutation in the AKT1 gene. It's sporadic and mosaic. It's not inherited in a sense, since it happens randomly during the early stages of embryological development, and all of the daughter cells born from that originally mutated cell become affected. Those tissues could involve the CNS, it could involve the feet and extremities, hands, it can involve parts of the bone, muscle, skin, or fat in the abdominal cavity as well. So the presentation is quite varied, but there are some things that are highly conserved. For example, the CCTN lesions on the foot, and you can see on the hand here, are almost universally associated with this disease, and if you see it, are in fact pathognomonic for Proteus syndrome. So PROS, again, related but distinct constellation of conditions resulting from a gain-of-function mutation in the PIK3CA gene. There are some differences in the types of mutations. In other words, there are different mutations within that gene that can give rise to different phenotypes, but they're all gain-of-function mutations which give rise to hyperactivated PI3 kinase, which cause increased signaling along that axis, after which AKT inhibition should be successful. So you can see on the left, a lot of these diseases were identified in the past, and they were described sort of phenotypically, like CLOVES syndrome, which is an acronym for the symptomatology, or in many cases they were named after either the patient or the physician who described it. It was only fairly recently in the last few years that this genetic cause was identified, and so that's why they've all kind of come back together and are identified collectively as PROS. It's very important that this sort of unifying condition has been agreed upon, in particular with the FDA, since our trial that we're going to be conducting, a phase 3 registrational trial right now, is for PROS. It's not for one condition or the other. The fast track designation that we received from the FDA last year for this condition was for PROS. It was not for one of the conditions or the other. Again, this condition collectively is much, much more prevalent than Proteus itself, at least 10 to 20 times more prevalent. So here's a high level of our registrational trial designs. We're there on the left, starting these 2 registrational cohorts right now. These are open label responder trials. So every patient will either respond or not, depending on their performance to predefined criteria, and some sort of threshold will be discussed with the FDA about the merits of approval. So the first cohort will be Proteus syndrome only. The second cohort will be PROS only. And the third cohort will be for those patients with either Proteus or PROS who do not qualify for whatever reason based on the exclusion and inclusion criteria for one of the first 2 cohorts. So moving now into our second generation AKT inhibitor, ARQ-751, which is designed to be much more potent and selective and have a wider therapeutic window than our first generation moransertib, which of course we're progressing now mostly in orphan conditions. So as Pete mentioned, ARQ-751 is currently in a phase 1 to 2 solid tumor trial in patients who harbor an AKT or a PI3K or even a PTEN null mutation. So it's very well known, and it's been tracked by many other large pharmas who have come before us, that 3 to 5% of solid tumors, regardless of the originating organ, harbor these AKT or PI3 kinase mutations. It's not surprising, since it's a pro-growth axis and anti-apoptotic axis, that this would be the case. But still, it's a very common condition in many tumors, especially breast, colon, brain, etc. So here's our phase 1AB, phase 2 trial. Now that we've picked a recommended phase 2 dose of 75 milligrams, we can go forward with this program in phase 2, as Pete mentioned, most likely in breast cancer, but there are certainly several other solid tumors for which this has great potential. very quickly on our finances, we ended the year with roughly $100 million as per guidance, and we expect to end this year with $60 to $63 million. So we're very well capitalized and lots of opportunities for non-dilutive funding as well. That sort of concludes the prepared remarks. Happy to open it up to questions for Peter or myself. Yeah, no, absolutely. It's a great question. I'll just repeat it for the audience. So how much BTK inhibition do you need to be a successful BTK inhibitor? I think what we've seen, and you can compare it to maransartib in Proteus and PROS is a good sort of contrapositive. You need profound suppression of the BTK signaling axis around the clock 24/7, I think, to see clinical activity. You don't want to turn down the dial. You want to completely obliterate or abrogate the signaling of that thing. And we've seen, not even referring to our own molecule, although certainly there's emerging clinical evidence that is operative with our molecule as well, but you can look at approved molecules with many years of experience like ibrutinib, that is also the case. And in fact, ibrutinib has good receptor occupancy at much, much lower doses than what's commercially approved. So I suspect that they felt that once you had good receptor occupancy, that was not sufficient. They wanted to go even further, because when we test phospho-BTK, and now we're seeing across the board 100% inhibition at the doses we're at, that's not where you stop. We saw that at around 20-30 milligrams, and we're now double that and maybe triple the exposures. So that's really where we think that we're going to get the optimal efficacy, and certainly that's what ibrutinib did as well. The other thing I would mention is that when we do our phospho-BTK assays, it's not a perfect assay, and it's at a point in time. It's at a snapshot in time. And we know that we want around-the-clock suppression to prevent these cancer cells from escaping, not just 100% at a random time point. And so that's why I think it makes sense to not cut it too close, if your safety will allow, to not cut it too close to the bone. Again, I mentioned maransartib in Proteus and PROS. That's a different story. So we're at much lower doses than our cancer doses in this case, because we're not trying to completely obliterate the AKT signaling in these young children who are normally trying to grow or going through puberty and are actually, there's some normal growth that we want to occur. So in that case, you're turning down the dial, if that makes sense, as opposed to obviously trying to kill a cell, but also trying to completely abrogate the signal.
That is actually very interesting. That kind of leads into my next question, which is basically ibrutinib has an incident online. And what happens is you basically get the turnover of the entire cell until there's a BTK form, which makes me wonder, why do you need a dose that's higher than 100% occupancy?
Yeah. Well, I'd say it's not infinite. So the lifetime of a BTK molecule is probably about a day. So when ibrutinib binds to that molecule, it probably stays on for the rest of that kinase's lifetime, which is about a day, much less than the lifetime of the cell. And so that's in a constant state of flux. So the enzymes are being chewed up, and the enzymes are being created anew by the cell. And so you need to have a constant replenishment of ibrutinib to continue to inhibit the newly created kinases. And so it's not infinite. It does need to be continuously dosed, just like our drug needs to be continuously dosed in such a fashion that the concentration creates inhibition of all of the kinases in that cell at all times.
Are there issues along those lines more?
Yeah, absolutely. No, it's a great point. The question was around finding patients for Proteus and PROS, and I'd say yes. I mean, absolutely. On the one hand, it's with any ultra-orphan condition, that's one of your hurdles. I would also say, I would add another hurdle, which is finding the right patients, because we know now from our clinical experience what are the types of lesions and what are the types of patients that are most likely to respond to our drug, because we've seen them respond. So we really want to, especially with PROS, which is much, much more common and it's much easier to populate the trial, we want to populate the trial with the right patients, not just get 20 patients in as quickly as possible. We'd rather take an extra month or two and have the best patients in there who are most likely to respond. That's the need for that third cohort, right? Because we want to still be able to provide drug to folks who don't qualify for the criteria, and we'll follow them and look at all the functional endpoints, et cetera. In terms of Proteus, I think one of the other mitigators is that we've been working hard globally to identify sites, areas of sort of academic excellence, genetic centers, and we've identified them here locally at the NIH and in the U.S., but also in Italy, in Germany, in Australia. So we have a lot of, we've done a lot of footwork already, legwork, to identify these patients and the sites that are magnets for these types of patients. It's actually pan-ethnic. There's no real founder population. There are certain rare conditions. If you look at like TTR amyloidosis, for example, it's autosomal dominant, and it clusters in these families. Familial AD is fairly rare. It's autosomal dominant, so it clusters in certain, like in the country of Colombia, randomly there's a bunch of folks with FAD. This is not like that. It's pan-ethnic. It's spontaneous. It's non-inherited. So they're everywhere, and we've actually identified, we need 10 patients for our Proteus trial. We've identified already 37. So we've identified them. They're out there. We want to get the right patients. One of them, I was just, as an example, is in Iraq. So, scientifically an ideal patient, but complicated. So we're going to run into some of those issues, but we'll take the time to do it right, and we'll enroll the right patients.
One last one on your program. Are there any lingering concerns in the community about the PI3K pathway for the target? I know there was some people studying.
For which program?
For the PAM program.
PI3K or BTK? I mean, I think, at the end of the day, clinical data trumps all. So if there are concerns related to safety with AKT, I mean, of course, there are class-related effects that will be present in any AKT program. And so the question is, what is your window, and what is the patient population, and how are you dosing? What is the accumulation? The devil's always in the details. So with respect to orphan, it's much lower doses than has been explored previously, and we've seen even with chronic dosing, a lot of our patients more than a year on therapy, including one patient who's getting a much higher dose because unfortunately she has Proteus syndrome, but also an ovarian cancer that's AKT driven. So we feel very, very confident with our therapeutic window in children, even chronic dosing in children, given the much, much lower doses. In cancer, of course, we feel very confident because we have such a potent and selective inhibitor, but at the end of the day, it's clinical trial data, a mountain of clinical trial safety data that's going to give us the utmost confidence, because that, of course, trumps everything. Thank you.